All right, well, let's go ahead and get this thing started. So first of all, welcome, and thank you all for tuning in to today's webinar. We're very excited to be able to learn more about the exciting work that Dr. Ghazaleh Sadri-Vakili and her team are doing to identify new treatment targets for ALS and other neurodegenerative diseases. Thank you as well to the rest of my team at Unite for working behind the scenes to make this event happen. My name is Tanner Dagdalen. I'm the co-founder and chief executive officer of Unite Genomics. And at our company, through the technologies that we're developing, we aim to rapidly increase the research community's understanding of ALS, get more patients into trials faster, and cut years off of the discovery cycle time for researchers. Our team envisions a world where all patients are empowered to improve their health and no patient's disease goes untreated. And for us, this world is 100% possible. And as a community, we can make it happen. So in this event, Dr. Sadri-Vakili will first be presenting for about 30 to 45 minutes about the current research underway in her lab, after which the two of us will have a follow-up discussion and some Q&A. So if you have a question at any time during the event, please use the Q&A feature to submit your questions, and we'll reserve time at the end of the session for a Q&A discussion. So now, without further ado, let me introduce our speaker. Dr. Sadri-Vakili directs the Neuroepigenetics Laboratory at the Mass General Institute for Neurodegenerative Disease, where she focuses on identifying common mechanisms that cause neurodegenerative disease to identify novel therapeutic targets. She's also an assistant professor of neurology at Harvard Medical School and a scientific advisor for the Healy Center for ALS. We are honored to have you here, Dr. Sadri-Vakili. Thank you for joining us today. And if you're ready, you can go ahead and start your presentation.
Well, thank you for inviting me. I'm delighted to be here with you today and share some of the latest work that we're doing in the lab. Let me know if you can, let's see. Does that look okay?
Looks great.
Okay, great. So yes, my name is Ghazaleh and I'm delighted to be here with you today. Thank you for inviting me. I always love to meet with patients, patient advocacy groups, and learn from you guys as much as you think you're learning from me. So I thought it'd be helpful to start by just kind of talking about how do you go from an idea to getting a drug to patients. And this is really a really crude way of talking about this, but really you have an idea. We work on some fundamental research. That's really what my lab is focused on. Maybe we can do some preclinical testing of a compound to then, if we have positive results, to get them into clinical trials. As you know, there are 3 different phases of clinical trials, followed by regulatory approval and patient care. So we come in at these points where I've put these arrows, the conception of an idea, and then doing molecular, biochemical, and cellular assessment of postmortem samples from patients, and then identifying novel drug targets that then we'll use our cellular and animal models to go after. We test a lot of novel compounds or FDA-approved compounds. And really, I'll tell you one story that focuses on that and another one where we found a target. We also work very closely with our colleagues at the Healy Center. I'm really lucky. I have really wonderful clinicians and other scientists that I work with at the Healy Center. And in this form, we use a lot of biofluids such as plasma or cerebrospinal fluid from patients, and we work closely with them to try to identify diagnostic and prognostic biomarkers of disease. Here's an overview of what I'll be talking about today. I'll tell you a story that you may have heard me talk about previously if you ever heard me give a presentation focused on inflammation and a drug called cromolyn sodium. This is something we've published previously. And then I'll talk a little bit about some of our new findings that's not published yet. We're actually putting together the manuscript now that we will be submitting in the next few weeks. And this is focused on a protein called tau, really well known to Alzheimer's, but it may be playing a role in ALS as well. Okay, so neuroinflammation is known as a pathogenic mechanism in ALS. We know that this inflammation is important for disease progression in ALS, and we know that because there's activation of the innate immune cells known as astrocytes and microglia. We also know that T regulatory cells are also the number of T regulatory cells is decreased. And there's an increase in pro inflammatory biomarkers in brain, spinal cord, and blood of patients. On the right hand side, I'm showing you a PET imaging study that was done in patients living with ALS compared to healthy controls using a PET ligand that recognizes inflammation, PBR128. And you can clearly see here that the red and yellow binding of the marker is a lot more in the ALS brain compared to control. So this is a way to visualize inflammation in a patient brain. So together, all these studies suggest that perhaps if we target inflammation, maybe we could mitigate some of the symptoms in ALS and stop disease progression. So one way that we could do this is to change the balance between what we're calling reactive microglia or astrocytes. And you can see that here, when astrocytes or microglia are activated, they release pro-inflammatory cytokines and chemokines that could then exacerbate inflammation. So what we'd really like to do is to take them from the reactive state, which is what has been described extensively in ALS, back to their homeostatic state when they're not pro-inflammatory. So if we could tip this balance, then perhaps we could provide some neuroprotection. So given that we know this, we'd like to decrease inflammation by decreasing the reactive state. We thought, okay, so what are some compounds that could do this for us? I just told you the hypothesis, and now I'll tell you that in my lab, we're really using a multi-pronged approach to understand the role of inflammation and also to target it. And these are all these different projects that we have in the lab. I'd like to tell you about them at some point, but because of time today, I'm just going to focus on one thing that we do, which is test novel therapies in our mouse models. Okay, so why do we think about chromalin and sodium? We thought about chromalin and sodium actually through our discussions with Dr. Rudy Tanzi, who's a world-renowned Alzheimer's researcher. And what they were doing in their lab is they have this 3D cell culture assay, and they were testing thousands of different compounds to see which one of them would confer neuroprotection in their Alzheimer's disease cellular model. And one of the top hits that came out of that screen was chromalin sodium, which is a mast cell stabilizer, and it's been shown to decrease the inflammatory response by decreasing inflammatory markers. And what they found after that is they treated Alzheimer's disease mice with chromalin sodium, and they found that it does exactly what I was telling you, that it took the microglia from the reactive state into a more homeostatic and neuroprotective state. So that was really exciting, and it sort of built the foundation for what we decided to do in ALS. And we hypothesized that chromalin sodium would be neuroprotective and delay disease onset and progression in the SOD1 G93 mouse model of ALS. I'm showing you the study design because I think this is really important. You have to use a really large sample size for the mice. These mice have a lot of variability in them. We use both male and female mice, and you can see how they were broken down into these different groups. We have wild type mice, which are the litter mates of the transgenic mice that have the SOD1 mutation. And all these mice were then treated either with vehicle control or they were treated with chromalin sodium at one dose that was suggested by the company AZ Therapies that provided the drug. Following treatment, we assessed the behavior. You can see all the different parameters here: body weight, neurological score, motor tests, as well as survival. The caveat being that this is not a real survival. It's actually when the animals reach a neurological score of 4. So that means when you put them on their side, they're not able to right themselves. This SOD1 mouse model has severe weight loss as well as paralysis. So those are some of the things that we look for when we're testing the neuroprotective effects of a drug. After the mice were euthanized, we then assessed their lumbar spinal cord motor neurons. These mice, again, lose a lot of their motor neurons in the spinal cord. We assessed inflammatory markers, and we also assessed the neuromuscular junction. So these mice also demonstrate denervation at their neuromuscular junction, and we assessed the tibialis anterior muscle for that. So what we found is that in mice that were treated with chromium sodium, there was a significant effect on paresis. It just means that these mice had a delay in their symptoms. So the mice start to have tremors, and then slowly their hind limbs are paralyzed. So it's at the point at which the mice start to show symptoms is what we call the paresis onset, and you can see that there was a significant effect on that. Also, there was an effect on the peak body weight. So the mice, as I told you, start to lose weight, but the ones that were treated with chromium sodium, they had a delay in their weight loss. We looked at motor performance. This is a grip test, and we found that at the later stages, so at postnatal day 120, we saw a significant difference between mice that were treated with chromium sodium. It had a significant effect on maintaining the grip strength, although it wasn't that noticeable, but still this was statistically significant. When we looked at survival in these mice, we didn't find a significant difference. Chromium sodium had no effect on survival. However, if you start to parse it out and look at females and males separately, there was a significant effect on survival in the female mice and not in the male mice. This is not really surprising. We've seen this in our lab many a times. These SOD1 mice in the females, their disease progression and onset of disease is actually slower. It's about 5 to 7 days delayed compared to the male mice. And so having a positive effect in the female mice and not having one in male mice has previously been reported by other groups as well as ours. Someone wants to fund this, I'd be happy to look into it more to figure out why this is, but this is just, something that we see often. Okay. So next, after the mice were euthanized, we wanted to look and see what is the status of the motor neurons. You can see these large cells are actually the motor neurons in the spinal cord. And not surprisingly, the vehicle-treated transgenic mice had significantly decreases in the number of motor neurons. But you can see that chromium sodium treatment had a positive effect and that it helps spare these spinal cord motor neurons. I told you that microglia are involved and that chromium sodium had an effect on the microglia, decreasing their reactivity in the Alzheimer's mouse model. So we wanted to know if it had a similar effect in the SOD1 mice as well. I don't think you need a microscope or anything to really appreciate that the transgenic SOD1 mice have a lot of microglia, as you can see here, compared to their wild type littermate controls. And unfortunately, there was no effect on the number of microglia that were activated following cromolyn treatment in our hands. We assess pro-inflammatory cytokines and chemokines, both in the spinal cord as well as the plasma. Here's the data from the spinal cord. Again, you can see that similar to previously published results, there is an increase in all of these markers. Actually, IL-5 is an anti-inflammatory, excuse me. But the ones that are pro-inflammatory, you can see that they're all increased in the transgenic animals, and that cromolyn had a significant effect on CXCL1 levels as well as TNF alpha, so a chemokine and a cytokine, and that was promising and exciting. When we measured the same markers in plasma, and actually we're using this assay called an MSD assay, so you can look at 10 different cytokines and chemokines at the same time, and you can see that more of them are showing up in plasma compared to the spinal cord. But what was really interesting is that we saw similar changes between plasma as well as spinal cord, where there was some significant effect of cromolyn, not across board, but on some of the ones that we've seen to be increased previously. Next, we assess the denervation that occurs at the neuromuscular junction, looking at the tibialis anterior muscle. You can see here again that in the vehicle-treated animals, there is denervation at the neuromuscular junction in the tibialis, and that cromolyn treatment was able to rescue this effect. Finally, we measured the amount of the number of mast cells as well as the activated mast cells. And what I mean by activated is that they're degranulated. So what we found is that although there was a significant increase in the number of mast cells in the transgenic animals, whether they were treated with cromolyn or not, there was a significant decrease in the number of activated or degranulated mast cells. And you can see that here. So when these mast cells are degranulated or activated, they release histamine and other types of pro-inflammatory cytokines, which will then cause more inflammation. And what cromolyn did was actually stabilize the mast cells and decrease their degranulation. And this is really important because mast cells are a direct target of cromolyn sodium. So that was really nice to see an on-target effect. Okay, so to summarize this part, I've shown you that treatment of transgenic SOD1 mice with cromolyn sodium delayed disease onset. It was neuroprotective as it spared spinal motor neurons. It maintained innervation at the neuromuscular junction and reduced inflammatory markers and mast cell activation. What was really special for me is that on September 9th, 2020, AZ Therapies, which is the company that provided us with cromolyn to test it in our mouse model, started their phase 2A study of ALZT-OP1A in ALS. And so they started a clinical trial based on our findings. And that was really exciting because if you come work in a lab, you'll see that a lot of times things don't work. So this was really special and exciting for us. And that's really a nice example of, starting off with an idea, doing the experiments, and then translating it into the clinic. So I'll stop there for this, and then I'm going to switch gears a little bit and tell you about something new and exciting that we're doing in the lab. These bubbles represent all the different types of pathogenic mechanisms that have been described in ALS or linked to ALS. As you can see, there are so many different ones. The next story that I'll tell you focuses on the ones that I'm highlighting here for you. And the point I'd like to make really is that these mechanisms are linked. Even the ones that I'm not highlighting, you can't have changes in one cellular process without really affecting something else. So it's really important to keep that in mind and know that a lot of things that we're measuring would also have effects on other cell organelles or other types of cellular processes. Okay, so protein aggregates, they are a hallmark of neurodegenerative disease. And you can see here on the left-hand side, protein aggregates have been described in Huntington's disease, Alzheimer's, Parkinson's, prion disease, as well as in ALS. And on the right-hand side, I have highlighted several genes that have been shown that the mutations in these genes in particular cause ALS. And again, on the right here, you can see motor neurons that have aggregates of SOD1, TDP43, FUS, and C9orf72, all genes that have been directly linked to causing ALS. Now, if you think about protein aggregation and all the different ways that it could disrupt cellular processes, here's one way. All right, so we have these, this is a really crude schematic of a neuron, and you can see these highways or microtubules that are really important. So just like you have this two-way highway or freeway if you're in California, this is exactly how the microtubules are, if we want to just think about them simplistically. What they do is different types of proteins can bind the microtubules and just travel down back and forth, moving cargo throughout the different compartments of the cell. So you can take stuff from the cell body and take them all the way down to the synaptic terminal, down the axon, or you can bring back stuff from the synaptic terminal to the cell body. There's also trafficking between the nucleus and the cytoplasm, but I'm not going to be talking about that today. So you can imagine when a protein is aggregated, like SOD1, then you can cause this type of a pileup on your microtubules. And this would then not allow you to be able to move this cargo from where it needs to go, from the point of origin to where it needs to go. So if you have these aggregates forming, then you can disrupt so many different things from happening. You can disrupt proteins from doing their job, interacting with the right types of proteins, and all of that, just like this pileup would. So I'd like to talk about this protein called tau, which I'm sure you've heard about in Alzheimer's disease, and it's directly linked to Alzheimer's. Tau is a microtubule binding protein, so it's sort of like one of these little cars, and it's important for stabilizing the axonal microtubules, and it's important for trafficking things around the cell. It's implicated in AD, and tau has been shown to aggregate in a number of neurodegenerative diseases. And those neurodegenerative diseases where you have this aggregation of tau and mislocalization are known as tauopathy. So another term that you may have heard about. So what we found is that if you take postmortem samples from ALS brain, the motor cortex to be specific, where the upper motor neurons are, and as you know, the upper motor neurons are can also be affected in ALS. What we find is if you fractionate the cells, so you take the whole cell and you homogenize it, or we take that whole cell, homogenize it, and we can spin it at different speeds, then you can fractionate the cells. So we can fractionate it so we have the synaptoneurosomes, which are the synaptic portion of the cells. You can have the cell body portion. You can have even the nuclear portion. I'm not showing you the nuclear portion here, but I'm showing you the total extract, the cytosolic fraction, and the synaptoneurosome fraction. So what we found is that tau is mislocalized from the cytosol to the synaptoneurosomes. Importantly, phosphorylated tau at serine 396 is also mislocalized to the synapses. And what's happening with this phospho-tau is really specific to the ALS samples when we compare it to controls. So, and this has been done in over 38 or 40 samples. So it's a really, really large sample size. Now, interestingly, if you can look at this schematic on the right-hand side, hyperphosphorylated tau at this specific serum residue has been shown to have many toxic consequences in Alzheimer's disease as well as FTD. You can see this here, right here. So when the tau is hyperphosphorylated, it will form aggregates, and those aggregates can then disrupt trafficking, alter mitochondrial function, and also oxidative stress, which is also another pathogenic mechanism that we talk about in ALS. So given all these things, we wanted to know what's tau doing in ALS, right? So we hypothesized that alterations in tau plays a role in mitochondrial dysfunction as it has been described in Alzheimer's disease. And we thought that perhaps targeting tau may reverse these deficits in mitochondrial dynamics and function. As you know, mitochondria are the powerhouses of the cells. They're very important for generating ATP. In neurodegenerative disease, such as ALS, we find that there is a decreased mitochondrial dysfunctional. They have decreased ATP production. They're not able to buffer calcium appropriately. There's alterations in mitochondrial dynamics, and I'll talk to you about it a little bit more. And that there's an increase in reactive oxygen species. That's why you might hear that maybe using antioxidants is good for you or neuroprotective. Okay, so my very talented postdoc, Dr. Tiziana Petrosiello, she took postmortem brain samples from ALS patients and controls. And the first thing she did is use electron microscopy to count the number of mitochondria and to start looking at the shape of these mitochondria. And what she found is that there is a decrease in the number of mitochondria in the motor cortex in ALS. But importantly, when she was measuring the size of these mitochondria, she found that there's actually a lot more smaller mitochondria in ALS postmortem brain compared to controls. So then we wondered, what's the mechanism of mitochondrial fragmentation or fission, given that we have a lot more smaller mitochondria in ALS? Mitochondria are actually dynamic organelles. They're actually very fascinating. So you can see here you have smaller mitochondria that can fuse together and create this larger mitochondria. And these larger mitochondria can then fuse together and form these large networks. And this is a fluid thing that's happening in the cell depending on what the environment is telling it. So larger mitochondria can produce more ATP, more energy. The smaller they are, the less energy they can produce. And actually, sometimes the mitochondria, if they're damaged, they can also be cut into these smaller mitochondria or fragmented. And this process is actually called fission, mitochondrial fission, when you're breaking them down into smaller bits and pieces. One protein that's important for mitochondrial fission is this protein called DRP1. And what we found is when we went back to our ALS brains, we found that there's a significant increase in DRP1 levels. And not only DRP1 levels, but phosphorylated DRP1 levels as well, which is the active form that does this fission process. So I don't have time to show you all of this, but we went through and we looked at our postmortem samples. I'm telling you, there's over 38 or 40 samples that we've looked at. We looked to see if there's a difference in tau and DRP1 in male versus females, and patients that had a limb onset ALS versus a bulbar onset ALS, brain samples that had TDP43 aggregates versus ones that didn't have. We had a few ALS FTD cases as well. And it didn't matter what type of tissue it was, as long as there was ALS as a diagnosis. We saw that the synaptic tau levels were altered in all of them, and DRP1 levels were also increased in all of those. So this kind of went hand in hand together across ALS subtypes. So then we wondered, okay, so are ALS synaptoneurosomes that are enriched in tau and DRP1 able to alter mitochondrial dynamics? Are we able to break down the mitochondria if we start treating with these synaptoneurosomes prep from ALS patient brains? And the answer is yes. So we took these cells, these neuroblastoma cells called SHSY5Y cells, and what we did is we treated them with either tau, which is this was a recombinant protein that we had in the lab, and either synaptoneurosomes prepared from control brains or synaptoneurosomes prepared from ALS brains. And what you can see here, looking at this cumulative frequency, because as I told you, the mitochondria are constantly changing shape. What we found is that treatment with tau or ALS synaptoneurosomes decreases the length as well as the volume of mitochondria. And here on the right-hand side, you can see we've measured the mitochondria and kind of parsed out the different sizes. You can see in the gray bars and the red bars, the two that we care about, there is a significant increase in the number of smaller mitochondria and a significant decrease in the number of larger mitochondria. We also looked at mitochondrial networks, and we found that the total number of networks is not different between these treatments. However, there's a significant decrease in the larger networks following treatment with tau or ALS synaptoneurosomes. Also, there's a significant decrease in the number of branches of mitochondria following treatment with tau or ALS synaptoneurosomes. So next we wanted to know whether targeting tau can mitigate this effect with the synaptoneurosomes. And we did something really exciting and cool. Our colleagues at Mass General Hospital, Dr. Steve Haggerty and Dr. Silva, they have created a novel tau degrader. So they're using this new this new way of creating these protein degraders, which is called PROTAC, or proteolysis targeting chimeras, where you're actually taking advantage of the cell's own trash removal system. There's two different ways you can remove the trash. One is through this proteolysis, the proteasome, and the other way is through autophagy and the lysosome. So this actually takes advantage of the cell's own proteasome and tags and brings the ubiquitin ligase to your protein of interest that you want to degrade. So what we did first is to see whether or not this their PROTAC, which is called QC01175, whether or not we were able to degrade tau in the cells before we did anything else. And the answer is yes, QC was effective at reducing tau levels. But more importantly, treatment with QC was able to mitigate these changes in mitochondrial length and volume in response to tau and ALS synaptosomes. And you can see here the treated cells are here in the filled bars. And if you look at the red here, you can see a significant decrease in the number of smaller mitochondria following ALS synaptoneurosome treatment, as well as a significant decrease following tau treatment. Now, in terms of the larger mitochondria, it didn't quite reach significance for the ALS synaptoneurosomes, but it was very close. But we did see a mitigation of the effects of tau when you degraded tau itself. Similarly, we saw a significant effect with the larger networks as well as the mean branch length, which was, again, QC was able to reverse those, and that was really exciting. So what does it mean if you have larger mitochondria? What's the functional consequences of that? We measure the reactive oxygen species, or ROS, using another microscopy assay. And this was really exciting because we were able to show that when you mitigate mitochondrial morphology using QC, you're able to reduce reactive oxygen species. And that was really, really exciting. And the data is here. You can see with the QC, you have a lot less ROS production, whether you're treated with tau or ALS synaptoneurosomes. Okay, so to summarize, I've shown you that phosphorylated tau interacts with DRP1 and causes these changes in mitochondrial morphology, causing fragmentation and the appearance of smaller mitochondria. That I didn't show you this actually, but we have data showing that this also causes increases in oxidative stress. That if you target tau and degrade it using this new PROTAC QC, then what happens is you form larger mitochondrial networks and you get rid of the smaller ones. And more importantly, you're able to decrease oxidative stress. And with that, I'd like to thank all the members of my lab, past or present. Without them, none of this work would really get done because I'm always sitting and writing grants. And our collaborators at Mass General Hospital, people who were involved with the Chromalin project and the funding that was provided through Project ALS. Here are all the people involved with the mitochondrial study and tau. Of course, our outside collaborators, as well as our funding sources, and more importantly, thanks to the patients and their families. And I'll stop there, and I'm happy to take questions.
Awesome.
I'll stop sharing, so there you go.
I'm just going to set it to gallery view so everyone can see. Okay. Thank you, Dr. Sadri Vakili. I personally have lots of questions. We have a few questions in the Q&A chat as well. I'll start with those. The first question is, they were curious as to why the initial study focused on SOD1 mutations rather than other mutations.
The
Could you speak to
Cromlin
that? Yeah, the
study,
Cromolyn
system.
studies. Yeah.
Well, this is not a really great answer, but that's the mouse model that we have in the lab, and we've been using it for the past 5.5, 6 years. We have thought about bringing in other mouse models, and like a TDP-43 or C9orf72 mouse. Um, haven't done that quite yet, but I would say that the SOD1, even though it's focused on SOD1 mutations, it's really nice because it has a robust phenotype with the paralysis and the loss of motor neurons. So it gives us parameters that we can measure easily, and that's really why we've kind of focused on that mouse model.
Got it. And switching gears here again to the new stuff. You mentioned that you found that QC decreased or that you saw a decrease in the mitochondrial network. And there was a question about whether or not that was found in during a clinical trial. And I'll actually broaden the question to be because I'm assuming it was actually in not within the context of a clinical trial.
Right.
But I do have a question around was it in human tissue, or is that still a mouse model tissue?
Right. So we use human tissue to induce the fractionation and fission of the mitochondria in a human-derived cell model. So these are not iPSCs or anything. These are SH cells. They're more robust and you can grow them a lot easier. So this is really our first pass of demonstrating that if you take brain samples from an ALS patient, you can cause mitochondrial disruption in a cell, right? So the next step for us is, and I've written a couple of grants that I hope will get funded, but what we'd like to do now is, now that we have these exciting findings, to see whether or not you can reverse this in a mouse, and also in iPSC derived motor neurons that our colleagues are growing, to see whether or not there was an effect. The one thing I'll say about the QC, our colleagues, Dr. Silva and Haggerty, have tested in iPSC-derived neurons from FTD patients. They're more focused on FTD and Alzheimer's in their lab, but those are the next steps to look at it before we can get it to the clinic.
Got it. And if I can bring us back to the initial part of the talk on the Cromolyn research and specifically AZ therapies, you mentioned that actually, a few months earlier, AZ therapy started dosing patients, their first ALS patients, with the Cromolyn-based therapy. So just a few questions that I'm sure is burning in the minds of our community is is that study recruiting? Is what sort of how can they get involved with that study so that they can accelerate that? Do you have any information about that?
I don't have like exact information, but I'd be happy to get that for you. And I believe if you go onto the Healy website or the Neil's websites, they have all this information, as well as gov clinical
Clinicaltrials.gov.
trials. Yes,
Yeah.
that.
Okay.
So I don't have the specifics for it, but it is recruiting. I don't think they've stopped recruiting for that. With the pandemic, everything just kind of got slower and pushed back, and it was really hard to recruit and close out the studies. So I would just look on those websites or reach out to AZ Therapies directly, and I'm sure they can give you more information about that.
Awesome. So the other thing that I wanted to ask about the Cromolyn sodium-based therapies. So you mentioned in your talk that Cromolyn seems to delay paralysis onset, as well as decrease the onset of decreased grip strength, at least in mice. So is it being explored as a preventative treatment or a prophylactic treatment, or?
So we didn't set up the experiments to really say whether this could be preventative or not. We started dosing the mice around P60. I know that there's newer studies coming, and we called that pre-symptomatic in the mice. So postnatal day 60, when the mice was 60 days old, and then we continued dosing them till the end of their life. Now, I know now that if you look at the compound action potentials at the neuromuscular junction, there are studies that are coming out suggesting that you start to see deficits in the innervation at the neuromuscular junction as early as P45, P50. So what we consider to be pre-symptomatic at P60, it's not, I don't think that's real. And I think with neurodegenerative diseases like this, things start a lot earlier than we notice. You only start to see things when someone shows up with symptoms, right? And I feel like at that point, you're already like pushing the limit and it's getting too late when someone is actually complaining about like their symptoms and what's happening. So with the mice, it's similar. So what we call symptomatic in the mice is around P90 when you actually start to see them starting to drag their hind legs, one at a time. So we didn't set up the study to start injecting the mice at P90 when they're actually symptomatic. So we were doing it when we considered them not to be symptomatic, and we saw these effects. I'm not really sure if you treat the mice at P90, what would happen then, whether or not you'd see some of the effects we saw. So that's one thing. The other thing is what we know about inflammation, it may not be a causative pathogenic event in ALS, but it does exacerbate things. We know that inflammation doesn't occur across every individual that has ALS. For example, we've done another study that we published looking at LCN2, which is another pro-inflammatory type of uh, molecule in the body involved in immune reactivity and immune response. And we saw that the levels of LCN2 were increased in bulbar onset ALS, which is a faster progressing ALS, and linked, so faster progressing ALS is actually linked to increases in inflammation.
Hmm.
And so therefore, I'm not really sure, who this is going to work for. I think if you have a more pro-inflammatory response, if you have inflammation that even if it's secondary to your disease, like if you're a person that has that going on, then maybe this is the thing for you and we need to prevent it before it gets worse. I know I went on and on and on, but I hope you kind of got
No,
something out of this.
yeah, this is great. This is great. So, just to kind of summarize it for everyone who's listening here, it sounds like, as you mentioned, it's not necessarily neuroinflammation is not necessarily something causative. So, it's not necessarily, targeting neuroinflammation with a therapy could slow it down once it's already started. But isn't something that will necessarily be looked at as a potential cure or preventative treatment. Of course, we don't know until we actually start trying it and try it in humans.
Right.
But the early indications seem to suggest that.
Yeah, it seems to me from Dr. Appel, who's really the leading expert in, inflammation and ALS, is that if you are someone who's a fast progressor, you have more inflammation. And inflammation is increased as the disease progresses. So those things together, we know for sure.
So, do you expect the future clinical trials on therapies that are targeting neuroinflammation will be focused primarily on fast progressors or fast progressing ALS cases?
I'm not sure, and I don't feel too comfortable saying anything about that, given that I'm not a clinician and I don't really know all the different ways that they stratify patients and try to get them into the trials. But I would say that, the stuff we do in the lab, if we find a good biomarker related to inflammation, as an example, then that could be something that could be used. If you're starting an anti-inflammatory clinical trial, then perhaps a simple blood test, or if someone has given their cerebrospinal fluid, they can like test it and see whether or not, let's say protein X levels are increased. And then that could be one way to stratify and know that, okay, this new therapy will really work for this individual because we know X is elevated or not. So thinking about it more scientifically, that's more of a science answer. And I think a clinician would be better able to answer that.
Of course, yeah. You mentioned biomarkers. It seemed like you were able to see some decent effects, at least in your models, on the specific biomarkers. Do you expect that those biomarkers will also kind of be brought into the clinical research when you kind of hand off your findings to the clinical researchers?
So actually, a lot of people have looked at what we measured are cytokines and chemokines, which are chemicals that are released by your immune cells. So astrocytes, microglia, immune cells of the nervous system, they release them. Macrophages, for example, that do the same thing that microglia do peripherally, they can release them, as well as mast cells that I talked about. So a lot of that work has been done in patients looking at plasma cytokine levels, plasma chemokine levels. But I would have to say that not two studies agree with each other. There are so many different types of inflammatory cytokines and chemokines, and we're not really getting a real picture that, let's say, IL-6 is always increased in ALS or TNF alpha is always increased across ALS. And I'll tell you the reason why that is because our immune system is very sensitive. And even if you eat, like your diet can alter your immune response, right? Or you could have a mild cold or, have COVID. Any of these things can alter it, but something as simple as the diet can also affect the immune cells and immune response. Some foods are more pro-inflammatory versus other ones. So I think this is why we have a hard time using those types of chemicals or markers as biomarkers because they can just fluctuate so much depending on what's happening with the individual.
Got it. So that's not to say that they won't be looked at,
No, no.
but may
I
not
mean,
be. Yeah.
people, there's lots of studies out there looking at plasma cytokine, chemokine levels, and I can think of like at least 20 different types of studies. I'm just saying that the consensus between all of them is not so great. This is why, even though we find these changes, in the animal models as well as in patients, nothing's been so consistent that we can say, okay, this is a new biomarker for us.
Mhm. And you also mentioned that the the cromolyn sodium drug, at least that's being put forth by AZ Therapies, was already approved in asthma.
Yes.
Does that so just for the community here, will that affect the speed with which clinical testing can complete, or if it is approved, the speed with which it can get approved because it's already been
So
approved in some other indication?
I think so, but I have to say that the formulation that was used for asthma, FDA approved, they therapies took it and they've changed it a little bit to make it get into the nervous system a lot easier. They've also created an inhalant type of a formulation as well that could be used. So I'm not really sure what the regulatory processes will be around there, but I would think that it's a lot easier than if this was a completely novel molecule or drug that was recently developed. You'd have to have a lot more safety, whatnot information. The other thing is they are actually reading out and closing out a clinical trial using cromolyn sodium in Alzheimer's patients. And from what I hear, and no one's been unblinded yet, they're working on analyzing the data. Again, with the pandemic, everything being pushed back. But what I hear is that you can really see separation between two groups. And I'm hoping that this is the group that was treated. So because they did this trial in Alzheimer's disease first, I think we're going to learn a lot more. And I think that will be really helpful and useful for getting things approved a lot faster once the ALS trial is over.
Got it. And do you think some of the data or some of the insights that we learn from the Alzheimer's trial will be helpful in designing the ALS studies or accelerating the clinical testing process?
I think so. I mean, I think we learned a lot from the Alzheimer's drugs. Since it's already been designed, I think they probably used a lot of that information that they had in hand, and that's why they were able to really um, push it forward so quickly and get it started. So I do think, just working in the neurodegenerative area, I also work on Huntington's disease, and I was more focused on that before I started focusing more and more on ALS. But, there's things that we did there and we learned there that I was able to bring to my work in ALS. So it's always nice to talk to each other and learn from each other.
Awesome. So I have one more follow-up question on the cromolyn studies that you did. You
Sure.
mentioned that you saw a pretty stark difference in between the genders of mice.
Mhm.
Is there, and that there's pretty good consensus within the scientific community that that's a real thing. Many studies have seen something similar. Is that something that, is there any evidence that that carries over into human tissues as well? Do you expect to see a similar pattern, or is this unique to mice?
So far, we haven't found any sex differences when we're looking at least the things we're looking at in the lab. So the mitochondrial differences, the tau levels, DRP1 levels, inflammatory markers that we've looked at, nothing's really popped out for us in terms of significantly being different between males and females. Um, but that doesn't mean that it's not different, or there's specific things that maybe we just haven't seen that, and we don't really see something so overtly different between female patients and male patients in terms of their disease progression, as far as I know. But again, maybe when you talk to my clinical colleagues, maybe they have noticed these differences that I just don't know about.
Great. Okay, so bringing us back to the new research, which you presented on towards the end of your talk, I have kind of one final question on that, which is you mentioned that targeting tau with a tau degrader seems to mitigate the mitochondrial length changes that you were seeing between ALS and control samples, especially in the smaller mitochondria.
Mm-hmm.
Um, and that it also reduces the oxidative stress
Yeah.
that you were seeing. So you mentioned you had some studies that are in the works. So what's the next step there? I mean, that seems pretty promising. And ideally, come a year from now, you'll have a new presentation referencing another trial that has just been kicked off, or something of the like. So yeah, help us understand kind of what is the next step in that line of research, and then I'll have another follow-up question.
Sure. So one thing I'm really excited about is this concept of PROTAC. So, you know, making these little chemical molecules that will then tell your own cells to degrade things that shouldn't be clumping up in your cell, right? So, as you know, for example, TDP43 is a protein that is mutated in ALS and also forms aggregates. SOD1 can form aggregates. So, one direction that we'd like to go in is to partner with our colleagues that I mentioned already and try to degrade as many of these proteins that form these aggregates. And, hopefully clearing those will then allow the cell to, go back to doing its normal type of function and not be so much under stress. Because you can imagine, these smaller fragments in mitochondria not being able to produce as much energy to meet the cell's requirements would be a huge burden on the cell, or you having these tau aggregates, clumping up your highways and not letting other proteins get to where they need to go. So, getting rid of these aggregates is one thing. Getting rid of, and there's so many different ones as I mentioned. So that's one direction. The other direction is, to kind of run with this QC compound, treat mice, and not just SOD1 mice, but treat mice and then see whether or not this could be neuroprotective. Like get rid of the protein aggregates and then see if that will help with the survival of the animals, their function, their denervation of the neuromuscular junction, so on and so forth. So those are the first two things I see coming up quickly on the horizon. And then partnering up with our colleagues that use a lot of iPSC derived motor neurons, astrocyte, whatnot, and see whether these types of degraders would be beneficial in their cell culture models as well.
Awesome. And so for those who are interested in following up, what would you say is the best way to learn more about this new PROTAC system?
So it's all coming to a head at the moment. Our colleagues are starting a company. I believe there's two or three other groups across the nation that are coming up with ProTech focus types of companies. And I believe people are looking at it in the cancer realm. I always feel like neurodegeneration is like a step behind from the cancer field. But I think if you just look up ProTax, then you'll get a lot more information. They're not as easy to develop, but I think people who have been successful with it, they're doing really well, such as our colleagues, Dr. Haggerty and Silva. So besides that, because I feel like all these companies are just startups or starting in an incubator and just getting a lot more attention. I don't have anything else to share about them at the moment because they're just a little too cutting edge at the moment.
Mm-hmm.
But perhaps next year, you're just going to hear more and more about them.
Got it. So I have one final question before we wrap up here, and it may put you on the spot just a little bit. So, you mentioned that you have some research projects that you're hoping will get funded. And, the one thing that's just truly amazing about the ALS community is how connected and active and powerful they are. And, I've seen just miracles happen in this community. So my question is, how can we as a company in the space, but also the community at large, how can the ALS community support your research, support the development of new treatments on an accelerated time frame? I mean, time is of the essence here, right? The clock is ticking. So, we'll do whatever it takes to make things go faster and faster and faster. So do you see any opportunities for, any resources to come your way through, maybe activities in the community or activism or something like that?
Sure. So this is a really great question. It does put me on the spot, but there's several things that I can think about. One, I think I'm very lucky because I work at Mass General and I'm part of the Healy Center. As the Healy Center started the master trial. They're testing, I believe now we have a fifth compound that's in the works. So that's just really exciting. But
So you're
what's
talking
really
about the
exciting,
platform trial, right?
yes, the platform trial. Yes. So what's exciting for me being there at the Healy Center is I'm a basic scientist. I don't see patients, right? So I'm focused on all the molecules and chemicals and whatnot. But what's exciting for me and really great opportunity is that I have access to patient samples. And I think it's really important to always start with patient samples. This is something I drill into to all my trainees' heads. Start with the patient. If we start with a mouse, when you knock out genes or knock in genes and do these things, then other things are going to happen that may or may not be directly related to the disease that we're studying. Those are all great models, so it's great. But so having the opportunity to have over 40 ALS samples in the lab from postmortem brains, or having the opportunity to have access to biofluids from patients allows us to really, really go after what's going wrong in a patient, which is really important. So I'm lucky to be able to do that. I'm lucky to be at MGH and tied in with the Healy Center so that when we find things like chromalin or other interesting compounds from a number of industry partners that we work with, then I have a direct link to try to get it into the clinic and have these wonderful clinicians like Dr. Sokovich, Paganoni, Barry, all of them who are really master at designing trials and really getting things going with, especially with this platform trial, which is really exciting. So all of that being said, ALS is more of a rare disease. And I apply for NIH funding as well as foundation funding, and it's really hard to get NIH funding. And, the NIH budget hasn't really grown over the past few years. And so any type of philanthropy or any type of funding that comes to my lab would be great. And it would just support these types of studies that we're doing, and that would just always be helpful. And it's the only way that we can do science. I wish as a scientist that, we could just have all this money to do the work that we do and just sit there generating ideas and pushing things forward. But the reality is that I have to keep applying for grants. I apply for 13, 14 grants a year. So you can imagine how much time that takes up for me to write these grants. So I think having philanthropic support is huge. And that just sort of opens you up to be able to do all kinds of stuff, use the most cutting edge technology and the best things that are out there that you can use to speed things up. So I think that just kind of sums it up.
Yeah, and I'll summarize it as well in just a second, just to ensure that everyone really has some takeaways of what we can all do to take an active role in this process. But
I'm sorry to interrupt you. One other thing I was going to say, and I stopped there. The other thing that I love to do and my lab loves is actually meeting with patients and their families and talking to them, because for us as scientists, I've gone to know so many different patients and families, and I love it and I cherish those relationships. But it's really important for us to speak to them, to just really hear from them what's important to them, what really bothers them, to be able to think about the disease more clearly when we don't get to see them in clinic. And everyone in my lab loves to meet with the patients, asking questions, and it really lights a fire in you and really motivates you and helps you really think clearly about what you're doing and to set up the best experiment. So that's another way that the community can connect with us and just come meet with us and talk to us. We would love to have you in the lab anytime.
Great. Well, I'll put you on the spot here again. If you're open to it, we'll share your email address
Sure.
when we share out this
Absolutely.
recording, so that anybody who's interested can get in touch with you.
Sure. And we have a Twitter, lab Twitter. We put our papers on there and other things. You can follow us there too.
Great. And I'll just kind of wrap things up here. So this concludes today's discussion, but thank you again for joining us today. A very special thank you to Dr. Sadri Vakili. So everyone who attended here will be receiving a post-event evaluation form in your emails, and we would appreciate it if you could complete it to help us improve these events in the future. We appreciate everyone's support and really look forward to all of us working together to advance ALS research and treatments. And just to summarize what we just heard about ways that we can get more involved, we heard about the impact that having samples and human samples and biofluids can have on accelerating the research path. We heard about the importance of industry collaboration between clinicians in academia and companies, between researchers in academia and companies, between companies, that fostering as much communication and collaboration as possible can help accelerate things as well. And lastly, we heard about some of the opportunities to increase the amount of funding that goes into ALS research and the importance of ALS nonprofits in funding ALS research. And so, I think for all of us, better supporting ALS nonprofits, continuing to be engaged with ALS nonprofits and helping them fundraise so that they can also contribute those proceeds to research studies is a big way that we can make an impact. And lastly, we heard about the importance of getting in touch and talking to researchers as members of the ALS community to share your experiences, to share any certain things that are important to you that they may not already know about, and to really foster an open dialogue between the researchers who are working day and night to advance new therapies and the patients who are clearly in need of them. So with that, as always, the time to act is now, and we hope to see you again soon.
Thank you so much.
Thank you.
Bye.
Bye, everyone.